water management country in the world. And we're at the Institute for Water Education, so this is extraordinarily fitting. Professor Pollack received his PhD from the University of Pennsylvania in 1968. And he's become a renowned expert in the role of water in nature. That expertise ranges over the entire scientific spectrum, from biology through medicine and into basic physics. He runs a Pollack laboratory at the University of Washington, uncovering some of nature's most deeply held secrets. He's editor-in-chief of the scientific journal Water, a multidisciplinary research journal that brings together water-oriented research from all kinds of diverse disciplines. Professor Pollack has earned many distinctions, medals, and honors. I'm not going to read you the list, because once you hear from him today, you'll understand exactly how they've been earned. So Professor Pollack, please come up. Thank you.
Thanks, Tom. Being called an expert is really scary because it reminds me of an expert. Right? Anyway, okay, so I want to talk about water. And let me start by suggesting that, most of us presume that everything there is to know about water is already known. After all, water is the most common substance on the face of the earth, and it's so simple that by now, naturally, we know everything there is to know about the subject. And I'd like to start by proving to you that we don't. So I'll show you a few phenomena, and then you tell me how you can explain them. Okay, so we see, as I was walking from the hotel today, I looked up and you can see something like this. And, it rises from the sea, and the question is that we mostly never ask, how come there's only one puffy cloud there when the water is rising all over? What is it that takes the water and brings it almost magnetically to this place? I'm not sure if you ever asked the question. Another question is, when you drop water on water, droplets of water, you expect that they will combine instantly. And actually, it doesn't always happen. We have some experiments here in which we show that you can take droplets and drop them water on water, and they don't coalesce instantly. How come? Another experiment, a rather dramatic one, is you take two beakers and you stick high voltage and probes into the two of them. You turn on the voltage and look what happens when you do that. So it forms a bridge, and if you take one beaker and displace it to the left, as you see here, this bridge sustains itself. And it could sustain itself indefinitely, and it could sustain itself over a distance of something like 4 centimeters or so. And so how come this occurs? And those are just a few questions among so many questions that still arise, and we really don't understand the mechanisms underlying these phenomena. Well, we started getting interested in water actually about a decade ago. I used to do muscle contraction before that. And I wrote a book about 12 years ago called Cells, Gels, and the Engine of Life. And the book was oriented toward biology. It was oriented toward, well, what's the role of water in biology? Now, you all know that you're two-thirds water, approximately two-thirds water. As you get older, you have less water. But that two-thirds translates, if you think about the fraction of water molecules instead of volume, because the water molecule is so small, it turns into more than 99% of your molecules are water molecules. I don't know if you ever thought about it. So you're essentially a bag of water molecules. And one of the things that came from this is, number one, that water is absolutely central to everything in biology, contrary, I believe, to what the textbook tells you, the cell biology books. But more than that, we realized that there's a kind of ordering that occurs. The cell is filled with massive numbers of proteins. They're all pretty close together, packed together. And right next to each protein, the water is ordered. And we tended to draw it this way, where the water is represented as a simple dipole, a water molecule. And they would all kind of line up into some sort of ordered fashion. But we didn't know how far out this extends. And that was the question. But one of the features of this water that we came to, others had come to the same conclusion, is that this water is in this region here, it's almost crystal-like, and crystals like ice tend to exclude solutes. And that seemed to happen inside the cell, that the regions of water next to the proteins would tend to exclude solutes and particles. So we were looking for an experimental model that we could use to see, well, how far does this extend? What are the features? This was just a preliminary idea of what happens. And we came upon something, and that happened about 10 years ago. And the idea was very simple, that we put a gel into a chamber, and then we added water and particles. For particles, we use these little spheres called microspheres. And the idea was this. The idea is we put the particles or microspheres in and see if they're excluded from near the surface. That would be the ordered water region. See how far they're excluded. Because if it's this distance, then we could surmise maybe that the ordered region extended by that much. So this is one of the first experiments that we did. And it looked something like this. So these particles were pushed out, and they're pushed out for a really large region. This would be roughly, we're talking 50 or 60 micrometers. And if I continue the video, then you can see them dancing around in Brownian motion. But they would never enter back into this region. Now this would be, if true, this would be astonishing, because we're talking about distances, huge distances by molecular standards. So we were very intrigued. We began to see it again and again. And we called it, because it excludes I'll show you later, most everything, we call it the exclusion zone, or EZ for short. Easy to remember. So, okay, it's morning. Need to wake up. Okay, now I'll give you just one more example of, another example is a piece of Naphion. Naphion is a polymer. It's like Teflon, but it has charge groups, sulfonate groups. So it comes in sheets, really convenient. Take a pair of scissors and cut the sheet into any kind of shape that you like, plunk it down in the chamber, and add. But anyway, what happens is you'd see this region, the microsphere, the boundary is here, and this region opens up, and you get an exclusion zone, sometimes 400 or 500 micrometers, even larger than what you saw before. And so this exclusion zone is very prominent, and it happens with a lot of samples. And I want to tell you that the basic finding now has been confirmed by. Well, this is an old slide, by countless groups around the world. In fact, we found out it was published in 1970 by a group of physiologists. We discovered this after we found it. So that's not an issue, is whether it exists. It does exist. But what we'd like to do, what I'd like to present to you now, is getting to the basis of what's going on to demonstrate this, I think, a rather impressive phenomenon. The question I want to answer is it really general, as I've alluded to? Does it really arise from the ordering of water molecules? Because we're talking about huge numbers of water molecules that would be organized or ordered. If it's really the ordering of water molecules, then can that explain those first 3 slides that I showed you and more? Now, if it's ordering, you need energy to create order. So what energy is responsible for creating this kind of order? And might these findings apply more broadly in nature? So that's what I want to talk about. And the first question is generality. And to make a long story short, because I have a lot to present, we tried numerous surfaces. We tried various gels and polymers, biological surfaces, and monolayers, that is single molecular layers mounted on a gold substrate. And so long as the surface is hydrophilic, that is water-loving, which means if you have a surface, you put the water on it, spreads out, it doesn't bead up, then we find it. Now, when it's hydrophobic, then we don't find it. And among the solutes or particles, whatever, that are excluded, we have evidence, we've tried many of them, and it seems that this zone of water, this EZ, excludes particles and molecules down to 100 molecular weight or lower. We don't know how much lower because it's difficult to find out. we think it's down very, very low. Now here's just one example of what's excluded. And so what this is a piece of Naphion lying at the bottom of the chamber. And all we've done is we added water, no microspheres, and pH-sensitive dye. I think some of you know the pH-sensitive dye is like litmus paper. You stick it in, it measures, changes color depending on the pH. So this comes in liquid form, put a few drops in. And you can see all these pretty colors here, but that's not the point. The main point for this slide is that this is a mixture of different dyes, molecular weight roughly 100. And this region is clear. It excludes the dye. You can't see any indication of a dye here. So we know that molecular weight roughly 100 or so, the molecules are excluded. But I want to come back to this slide in a moment because the colors here, it's not only an artistic contribution, you might say, but the colors are interesting because this region, this red color, for this particular set of dyes, red color means pH of 3 or less. In other words, very, very acidic or full of protons. And that's important. I'll come back to it. Okay, so in terms of generality, many hydrophilic, not hydrophobic surfaces generate exclusion zones, and many solutes are excluded. So the bottom line, this is very general. Now, is this zone really different from ordinary H2O, bulk water? I've alluded to the possibility, but I haven't presented any evidence. And now we have a lot of evidence by this time. And I'm going to list the evidence. I'm not going to go through each one because otherwise I'd use up all my time. I want to just show you, demonstrate one of the 8 or 9 different ones that we have. Most of it's been published. So the first is that the easy water, that is the exclusion zone, the water molecules are more constrained than bulk water molecules. They're more stable than bulk water molecules, and this is the method that was used. Again, it's mostly published. It has negative charge, unlike bulk water, which is neutral. And I'll show you that piece of evidence because it's critical for what I want to show you afterward. It absorbs light in the UV region at 270 nanometers, and bulk water doesn't do that. It's more viscous than bulk water. The molecules are aligned in this region, and the molecular structure is different from ordinary water. And the optical properties differ, and that was actually found not by us, but by 2 Russian groups, both from Moscow, who don't even know each other, pretty much, and they published essentially simultaneously using different preparations showing that the index of refraction was 10% higher this water than for bulk water. Up to 10%, actually, up to 11%. Now, what about this negative charge? That's important, so let me just go and explain to you how we discern that. So what we did is we put, this is a chamber, and inside means inside of a gel. So this is a gel, a polyacrylic acid gel. That's what this is here. And outside means outside the gel, or we put water here. So gel, water. We take 2 electrodes. These are microelectrodes that taper to a tip used in biology for more than 50 years, less than 1 micrometer. So you have good spatial resolution. And we put one out here somewhere, a neutral electrode. And the other one's a probe electrode. It moves from point to point, and we want to measure the potential difference between these 2. So you can see with the polyacrylic acid PAA gel here, the potential difference out here between here and here is 0 at first, and then you get closer and closer. And once you get a few hundred micrometers from the interface, then you begin to pick up a negative potential. And this negative potential, the region of the negative potential corresponds to the region of the exclusion zone. So it looks like the exclusion zone is negatively charged. It's not neutral like water. So then we rip out the gel and we put a piece of Naphion here and we repeat the same experiment. And the result is shown here. And you see that the negativity is apparent, but it begins farther away and it gets more negative. Now I didn't show you because the slide didn't work, but the Naphion has a bigger exclusion zone, maybe twice the size. So again, the region of negativity, whether it's gel or Naphion, corresponds to the exclusion zone. So it looks like the exclusion zone is negative. Now, I know you're not thinking yet because it's too early in the morning, but if you think about it, what I've done in this experiment is I've taken water, which is neutral, and I put it in, and suddenly I'm suggesting to you that you get a big region of negativity. Now that doesn't make sense at all, because how are you going to take neutral water and suddenly create a big negative zone? Well, the only reasonable way that might be possible is that the water molecule is split somehow. So the negative ones are here and the positive ones are, somewhere. And the question is, well, is there evidence for the corresponding positive ion somewhere else? Well, I've shown you the evidence. This slide you've seen before, turned at 90 degrees to correspond to the previous one. So again, here's the naphthalene, here's the exclusion zone, which I showed you is negatively charged. And remember, you have a lot of protons here, so you have negative, positive. Indeed, there is looks like a splitting of the water. Now just to make sure that we're not fooling ourselves, we put one electrode here and one electrode here with a resistor in between to see if there's current flow between the positive and negative. And you can see that there is current flow. It starts fairly high and then goes down to, not to zero, it goes down to some plateau value which extends out in time. So it means there really is current flow between plus and minus, and there really is indeed a separation of charge between those two zones. So what it really means is that you have a charged battery in water. Okay, and yeah, you don't expect a charged battery in water, but essentially that's what we found next to hydrophilic interfaces only. So I've shown you that the EZ really does have negative charge and corresponding positive charge afterward. Now if you go back at this listing of evidence and just look at the blue ones to try to figure out what they mean in terms of structure of this zone. So the molecules are aligned and they're stable and they're constrained. And that's the best way to interpret that is some kind of liquid crystal. Okay. So we don't know what, I'll go into that in a moment, but to summarize so far is what we started with, and we found that indeed there's some kind of liquid crystalline structure that has negative charge and it excludes solutes profoundly from this region. There's some hint that I've given you that it's non-dipolar. I'll go back to that in a moment. It's not just this stack of dipoles that you see here. And it may extend very far. So by very far, the textbook says 2 or 3 molecular layers of ordered water next to various surfaces. We have 2 or 3 million molecular layers next to these surfaces. It was suggested 100 years ago that water might actually have a fourth phase, not the ice, liquid, and vapor that we all know about. And I don't know if this is fourth phase, but it's certainly different from liquid water. It's not solid. It looks somewhere in between, and there's a lot of it, and it's bounded. So now, if we're talking about non-dipolar water, so the question you're faced with, or we were faced with, well, gee, if it's not just a simple stacking of water molecules, then what might it be? So we scratched our heads a bit, and we thought, well, it has to be non-dipolar because the main problem is it's got negative charge. No, dipoles are neutral, so you can stack dipoles, from here to the moon, and you'll never get negative charge, right? Dipoles at plus minus, and so that doesn't work. Unfortunately, the book that I wrote 12 years ago suggested the dipole model, but we have to admit we were wrong. It's got to be something else. Now, so you need something that's negatively charged, some structure, and also this 270 nanometer absorption that I mentioned to you on this long list. That implies, usually the chemists take that as meaning some kind of ring-like structures. Dipoles are not ring-like structures. So those are a couple of the main reasons why we have to abandon this dipole, just stacking of water molecules, and go somewhere else. So where do we go? Well, we thought one way to go is to start with a structure of water that we already know is correct. And the one that we know is correct is ice, because there's lots of X-ray diffraction on ice. So we thought, well, maybe if we start with ice, maybe there's some variant on ice would explain, not ice, but the variant would explain this exclusion zone. And that would have precedent, and that would be a good place. So if you look at ice, look something like this. Now, these are two different views of the same structure. So the red ones are oxygens. You can see they form a kind of hexagon. The hydrogens are not shown. They're here, 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 and so on between them. And it's not exactly, it's almost planar, honeycomb sheets, but they're actually corrugated somewhat because of electrostatic forces. Now, if you look at this one, just a slightly tilted view, you can see these blue dots. So what are the blue dots? Well, the blue dots are protons. And what they do is they interconnect. So the oxygens are negatively charged, and the blue dot's positively charged. So basically, this forms a glue holding together these two negatively charged oxygens, and that's why ice is hard. So we thought, if we start with this structure, is there some modification that could explain the exclusion zone? And we thought, well, the first thing that's sort of obvious is, well, let's pull away these protons. If we pull away the protons, then we go from a neutral structure, ice, to something negative. And so we thought of doing that. We got the negative charge, if you can see it. And it's not solid anymore because you've taken away the glue. And so we thought, this is cool. And then we realized it's impossible. It's impossible because if you remove these protons, then you have negative opposite negative, and the structure would just fly apart like that. So it's a nice try, but it's unstable. So we lose. That was disappointing. But ice seemed so attractive, and we scratched our heads, and then suddenly it came that all you need to do is to shift slightly, to take the plane closest to you and shift it in one direction, and then you have something that's really attractive, because then you have a negative oxygen from the plane behind and a positive hydrogen, proton, in the plane closest to you, and plus and minus stick together. So you have many of these places where you have this stickiness, this electrostatic stickiness. Now, it's not the same as ice. It doesn't give you hard, but we don't want hard. This would be a kind of a weak connection that you could actually overcome with a little bit of shear, sort of gel-like. So that looked interesting. Now, so the way, if this is our kind of starting point, and then we thought, well, the way this could work is that you start with a hydrophilic material in water, next to water, and what happened is these layers would form one by one, and once you have a few of these layers, the next one would form from the water, would create the next layer, and from the water the next one, and so on. And if you looked at one of these, the structure would look something like this. Again, similar to ice. Here you do show the protons in between. And it's interesting because if you count in the unit cell, if you count the number of oxygens and hydrogens, it's not H2O anymore. It's actually H3O2. It's negative. There are more oxygens than there are hydrogens. That makes sense because it has a negative charge. But it's actually, it is correct, it's a chemical change. It's not just a rearrangement, it's a chemical change that goes from H2O to this kind of structure. A colleague just last week calculated the amount of energy that would be required to do this. It's very, very small. Now, what I've done is I've taken the plane behind and the plane above, and I've shifted it to the right. But I didn't have to shift it to the right. I could have shifted it to the left, gotten the same result, or 60 degrees, or 120 degrees, and you get the same. So the point is, for this conference, it's symmetry. This is symmetrical. It doesn't matter which way you shift. And that's important because of that feature. It gives you a kind of flexibility, structural flexibility. I mean, not in a literal, a figurative term. And you can get interesting structures if you start stacking these. And one structure looks like this, a helix. So here's layer 0, and here's layer 1 shifted this way, and 0 degrees, 60 degrees, 120 degrees, and so on. And with that, you can build a helix. So why is a helix important? Well, helix is important because in biology, almost all of the structural entities, the proteins, and DNA, RNA, are helical. And it's known that these helical structures have some kind of ordered water sitting right next to them, so you can imagine a DNA sitting right here, just abutting with water. So this is, you might say, an advantage of this. So summarizing the advantages of this non-dipolar EZ, there's precedent for it from ice. It has negative charge, which we want. The ring-like structures might be compatible to delocalize electrons with the 270 nanometer absorption that's observed. You can accommodate helical structures with it. And importantly, the large sheets eliminate the Brownian instability. Now, what do I mean by the Brownian instability? Well, I guess many of you know Brownian motion, you know, the natural motion, the wiggling around that occurs. And if you think of each one of these bricks as a water molecule, if you have stacked dipoles, in other words, stacked bricks, the first one is wiggling, the second one is wiggling some more because the first one wiggles, and so on. And by the time you get to the fifth or sixth or seventh, the wiggling is so great that you can never build beyond that. And so many chemists have argued that the idea of water structure, long-range water structure next to surface is impossible because of that. However, it's not what we're suggesting. We're suggesting, by the way, others have argued against that, but I'm just telling you. Now, so what we have is actually practically an infinite sheet, this honeycomb sheet. And this has larger mass, and because it has larger mass, the Brownian motion is reduced. Right? And so these are so large that the Brownian motion is reduced practically to zero, and the next layer is actually stuck to this layer, and so on. So you can build essentially indefinitely without having to worry that the Brownian motion will kill the structure and make it impossible. Now, so I've suggested reasons why the structure is not dipoles, but honeycomb sheets. Now, is there any evidence for it? Well, yes, there is. Not from us. Some investigators, actually from Harvard, published about 5 years ago, there's a protein subunit. It's a very old protein, ancient ATP synthase, and subunit C. Now, these subunits tend to come together in times of low humidity to form structures like this, or structures that tend to be geometric in shape. This is with the SEM and the TEM, and you can see them. And they did electron diffraction to see if there's any order of the volume of water that's contained inside. And this is a representative diffraction pattern. You can see that there's great regularity, otherwise you wouldn't see little spots. And you can see that there are 6 of these. So the authors concluded that the water, this is a volume of water, inside these volumes are ordered hexagonal sheets of water. This is exactly what we were suggesting. So the answer to question 2 is the easy, distinct. I think it's distinct, and it's a layered honeycomb structure, the best we can say. Now, does it explain some anomalies? This kind of structure? What behavior do we expect from a crystal as opposed to individual water molecules? Well, crystalline elements stick together, and so if you ask yourself, what about the gelatin dessert that you had last night, perhaps for dinner? It's 95% water. It's almost all water. Some gels I've handled are 99.95% water. That is essentially all water with a few strands of polymer stuck in. They hold their water, right? And the question is, why doesn't the water dribble out like your shower? You know, you lift up and the water doesn't come out. Well, I think the answer is, from what I told you, that this is a kind of a cartoon-like structure of a gel. It's got gaping holes, pores inside, and these pores are filled with water. However, the water is not just ordinary H2O. It's actually this liquid crystal that fills the inside. So you have a hydrophilic surface, and you have layer after layer. It sticks to the surface, and the layers stick to one another. Therefore, the water doesn't dribble out, even in gels that are 99.5% water. Another feature is that this stuff is gel-like. There's water. So when you ask yourself, when you feel the gel, and you say to yourself, well, gee, you know, this has this kind of funny gel-like consistency. How does that happen? The physical chemist will tell you that it has to do with the viscoelastic properties of the matrix. And I'd like to suggest to you that it might be due to the viscoelastic properties of the water. Now, another issue is the surface. And Hungarians will recognize this one as being an old Hungarian coin. And these coins and paper clips and pins and such heavy stuff floats on the surface of water. If you put it just beneath the surface, it sinks, but if you do it carefully, it floats. And the question is, well, how come? And we found in experiments that this crystalline water grows at the air-water interface. So there's something special. You think, well, it must be surface tension, but remember, surface tension, according to the classical explanation, arises from the top layer of water molecules. They can't the water molecule can't bind with the air, so this potential bond comes down and makes for an extra lateral bond, so you get some stiffer surface because of that. The question is whether one molecular layer is enough to explain what I showed you. So we started studying, and we found we just used a simple chamber with 2 pieces of glass and sealed at these edges, and you put in water and put in microspheres, so you can see that it's cloudy. So here's the air, here's the meniscus, and the cloudy region with microspheres. And we noticed that this top region, after 15 to 30 minutes, a clear zone opens up. In other words, and it's stable for 10, 20 hours. In other words, something like an exclusion zone exists at the top. And we measured the electrical potential of this region. We found right near the top it was indeed negatively charged, just like exclusion zone. And I'll show you in the next slide that this region is just like behaves like a gel. Or a thick rubber band that runs from one side to another. You can't really expand this zone right here as you would if it were just water. And I'll show you that in the next slide. So here you see basically the same slide as before. And the probe is going down, lifting up, and then moving from side to side. So it's perturbing mechanically the surface, and you see that this clear region remains the same height. So it's cohesive. So it looks like basically you have an exclusion zone sitting at the top of the water. It's not one molecular layer. It might be a half a millimeter or something like this. And I think that's the reason why the coin doesn't sink, and also explains something like this creature, which some of you know. It's a lizard from Central America, and this lizard actually jumps in the water and walks across ponds. Because it walks on water, it's called the Jesus Christ lizard. So you can see that question is one molecular layer enough to explain this, or is this thick layer that we talked about? And I think it's the thicker layer. And the same thing with the water dropping on the water. So I just showed you that the surface of the water is not just H2O, it's got this thick layer on the surface that's like EZ water. And this pendant droplet is also exposed to the air. It's also got an EZ layer. So when this falls, it's not water meeting water. It's EZ meeting EZ. And then you can understand why it might not be instantaneous. And what happens actually, it drops on the surface. It takes some time, and then boom, and does that. And it's like a series of dance steps. And this is known for 100 years. It's not new with us, and we like to do it because it's fun. Okay, crystals can be stiff, right? Salt crystals, sugar crystals, diamonds, what have you. And you can understand why, if you just have H2O molecules sitting in there, this is really hard to explain. But if you have a crystal that in some cases can be stiff, you can understand why this might hold together. And finally, in this series, an important feature is that this crystalline zone here is what keeps these protons from coming back in. These are actually hydronium ions, that is a proton stuck to a water. You have H3O+, which has some bulk to it. Now, ordinarily, if you have plus here and minus here, you know what happens, right? They want to do this. However, they don't do it, and the reason we know they don't do it is because we can stick electrodes in there a day after we set this up. Still, we get this potential difference. So the charges are maintained separated. And I think the reason is that this region here is so tightly packed, such a dense structure with a very, very fine mesh, that even though these want to come back in, they can't. So the battery charge remains separated. Okay, so the answer to question number 3 is yes. Liquid crystalline water explains many of the anomalies that we started with, plus more. And important is it explains why that water battery charges, why they remain separated. Okay, question 4. What charges the water battery? Okay, so, you know, your cell phone battery, the juice runs down and you have to plug it in to recharge the battery. And so, I mean, this is nature. It operates the same way pretty uniformly. So what is it where does the energy come from to separate this charge that ordinarily wants to come together and to create this order? Both of those need energy. And we found out that it's the sun. It's light that does it. The first indication of such was when a student came around. We had this chamber sitting here, and one of the students picks up a lamp and just for fun shines the lamp on the preparation, you know. And so we found that it didn't take a rocket scientist to figure out that, well, you know, you put the light on, it grows bigger. Maybe light is important in what's going on here. So then we started to do some experiments, and the first experiment is we used near-infrared illumination, that is 1 micrometer wavelength. So here's a control situation. Here's Naphion, here are microspheres. We used a green filter on the microscope, that's why it's green, just to reduce the microscope lamp effect. So it's actually pretty dark. Anyway, here's an exclusion zone and microsphere. Then we turned on the light, extra light, and we used, as I said, 1 micrometer turned it on 5 minutes, and you can see that this 5 minutes, the exclusion zone expanded by roughly 3 times. Then you take it away and it would come back slowly. And someone said, oh, you guys are stupid because you're using infrared illumination. Everybody knows that infrared causes heating. So this is probably an artifact of heating. So we measured the temperature at many points in the chamber at the end of this 5 minutes of exposure and never got more than 1 degree. So it is not due to heating. And then, of course, it was interesting to see what other wavelengths are responsible, not just 1 micrometer. So we did the experiment, and here is wavelength on this axis. So here's the visible region, here's the infrared region, and this would be the UV region. So, for example, we take a light, a green light, for example, and we turn on the green light, and the exclusion zone would expand by 60% or so. And we plot this point here and so on, try every different wavelength. So you can see the result. The wavelength really does matter. Surprisingly, the longer wavelengths, which have seemingly less energy, are the ones that create greater expansion. And in the infrared region, again, wavelength matters. And the most important wavelength is about 3,000 nanometers or 3 micrometers. Some of you may know that this is the wavelength that water absorbs most profoundly. So it looks like the wavelength that water absorbs most profoundly is exactly the one that expands the exclusion zone most profoundly. And this graph actually underestimates the real effect of this infrared because, a technical reason, the infrared light sources that we used here were 1/600th of the intensity as the light sources we had available here. So if we had used light sources that were at the same intensity, this curve would obviously be much higher. It would go way up here. So the real curve, if we had done the experiment with the same intensities throughout, would be some curve that goes up, and the highest point would be right here at 3 micrometers. So what does that mean? It means that, you know, visible light does count. It gives you some expansion, but the infrared counts much, much more. Infrared is what's most important for building this, is infrared. So now you might ask, where do you get infrared? Infrared is all over the place. It comes from the walls, it comes from you, it comes from me. If I were to turn off all the lights in the room and turn on an infrared camera, I get a great picture of everything in this room because it's all generating infrared. So infrared is literally, maybe figuratively too, free energy. It's energy that we get that comes, it's almost impossible to get rid of it, although you can. So the idea is that you have a material and you have water and some easy water forms, and then you get some additional, for example, infrared or other light, and this builds up more and more. And if you were to take it away, it would come back to its original. So, you know, having seen that you can take light, especially infrared light, and build up this exclusion zone, we asked the question, well, if you add light, you get bigger. What would happen if you subtracted light? So how do you subtract light? Well, it took subtract infrared. It took a few years for us to finally figure out how to reduce the incident radiant energy. Now, we're not talking about turning off the lights. We're talking about reducing the infrared light that's coming in. And finally, it occurred to us, all we have to do is go to Starbucks. And so, you know, if you put your cold drink in here, the radiant energy, infrared from outside, wants to come through, but it can't penetrate through. So we took one of these. Actually, we took a genuine Dewar from physics department laboratory, a bigger one and a professional one, and we put the chamber in to see what would happen. So we're now reducing the ambient infrared energy. And so this is the control. Here's the exclusion zone. 15 minutes inside the door, this is reduced by a factor of 2. 15 minutes out of the door, it's coming back to its original. So the answer to question number 4 about energy is easy buildup is powered by photonic energy, which orders the water, charges the water battery. It's sort of something like this. My son, the artist, he responsible for the book that came out of this stuff. Anyway, I'll show you a slide later. If you think about the possible energy flow in the universe, you have the sun hitting the water, and that generates heat, and you can go swimming in the summertime. But what I've shown you is that there's a different pathway, that the sun or light hits the water and imparts energy for building order and separating charge. Now, there may be 2 pathways, or another possibility is that there's one pathway and this degrades into heat. It's not so clear whether that's true or not. So if this is correct, if the sun hits the water and giving you potential energy, in other words, you have water and the light's coming in and gives you potential energy, you ought to be able to collect that potential energy somehow into useful energy. And so the question is, can you harvest this energy? Now I bet nobody in this audience has ever seen a glass of water doing work, right? But we found that it can. So it's not exactly a glass of water. One day, a student came running into my office to tell me what he found. He started with a tube made of naphthalene later, and it looks just like this. It's like a straw. It's empty inside. He had put this in the water, and the water actually contained some microspheres, and he was looking this way using a lens to see what's going on inside. And he came running in, and he told me, you know, there's flow through this tube, and it just goes on endlessly. It won't stop. So I thought, I almost fell out of my chair out of interest in this. And that's not expected, because if you have a tube, usually you need a pressure gradient to drive the flow through the tube. You can't get something for nothing. But anyway, it seemed that we were getting something. And the way the experiment is done looks like this. So you take the tube of naphthalene, you fill it with water, make sure there's no air bubble sitting in the middle, and then you take it and you put it into the chamber that has water with some microspheres, and put it in the microscope and see what happens. Okay, and what happens is that you see it looks just like this. Here's the naphthalene tube. Whoops. The exclusion zone here and here, and you can see that it basically just keeps running through. We've had this running now up to a day and a half without stopping. It just keeps going. We tried not only naphthalene tube, but we tried gel tubes. So here we tried 3 or 4 of them with success. This is a polyacrylic acid gel. We cut a tunnel through here by putting a wire in during the time the gel was forming and pulling out the wire. And then you stick the gel in the water with the microspheres. So what happens is you get an exclusion zone next to this gel surface, and you get one here. And this narrow region remains. And this is the region that contains the water and the microspheres. And what we found is, again, flow that just keeps going through there. And we also tried, more recently, adding light, because we think light is driving this. That's the energy, right? So we added more light, and we found that, it was just published, that we can get up to 5 times the velocity by cranking up the light some more, especially in the, yes?
In which direction does it run?
This is the 64 euro. We don't know. Well, we have a few hints. Sometimes it goes this way one day, next day it goes this way. Once it starts, we can turn the gel or tube around. It keeps going in the same sense relative to the gel. And we think it has to do with some asymmetry, if you will, because none of these tubes is exactly symmetrical. There's got to be some gradient, either of the energy that comes in, or the diameter of the tube, or some end effect, or something like that. We're currently studying.
Could you force it to get started in the opposite direction by blowing it and then seeing whether it maintains it?
If you want to join my laboratory, maybe we could use some people who think. Thank you. Okay, so anyway, so what I've shown you is that basically you put a hollow tube in the water and you get flow. Now this is work because the water has viscosity. So how do you get it? Obviously I think the perpetual motion machines are unlikely. You have to, if you get work done, you have to have some energy. So the system must absorb energy from outside, and I present it to you. We think it's light that's doing this. So the water is actually transducing light energy into mechanical energy. So you can harvest energy from a glass of water. Now, if you think about it, the plant that's sitting in your office is doing just that. It's taking the sun, right, and it's taking light, and it's converting the optical energy into chemical energy, right? And then from that chemical energy, the plant does everything. It metabolizes, it grows, it bends, what have you. And I'm suggesting to you that a glass of water does the same thing, that it absorbs light, and the light comes in, and it's not a surprise because this consists mostly of this. So there's not really so much difference between the two. And I like to say E equals H2O because I think you can't really separate the water from the energy. Water has energy. You like that equation? Okay.
Professor Pollack, you're in your last 10 minutes.
Okay. Yeah. Okay. Okay. So I think the last question may be foundational for any of all science involving water molecules and light. And what I kind of summarizing what I presented to you is that, in the textbook it says you basically have a particle or molecule in water, and maybe there are 1 or 2 layers of ordered water. What I've shown you is that there's a lot of this liquid crystalline water. It has negative charge, and you have positive charge around here. And all of this is driven by light. So this is vastly different from what you read in the textbook. And then the question is, well, if this is correct, what I've shown you is correct, we're talking about all chemical reactions. And so I think it might be necessary to return and ask the question of how do we really interpret all of these chemical reactions. So here's a question for you. If I take 2 of these, negatively charged, and I drop them in the water right near each other so that they can feel each other, what would happen to the distance between them? Now I know most of you will say, well, obviously they come apart from one another because they both have the same charge, but in fact they actually come toward one another paradoxically. This has been known for 100 years, and Richard Feynman was the one who coined the term like likes like. That is, they're like charges and they like each other, so if they like it, they want to come together. And he said like likes like because of an intermediate of unlike. The positive charge is pulling on the negatives. And so the question though has been, this is 60 years ago, where do you get these positive charges? I've shown you that they come from the buildup of negative charges here. It gives you positive charges here. So they do the attraction. And it kind of is something like this. You expect that these warring parties would not want to get close to each other. However, if you put the right and this actually, you can't see it here. It comes from the Tale of Genji, the first novel written in Japan. So anyway, they come together because of this, and you get stability when the attractive force pulling this way is equal to the repulsive force between these two. Then they're stable. And if you have, instead of 2, if you have a lot of them, you get something like this, which is called a colloid crystal. And a colloid crystal, well, basically the particles stick together because of this like likes like principle. And if you had yogurt this morning, probably the yogurt had a structure similar to this, and that gave the yogurt its consistency. And this principle, of course, could be used, it's basically self-assembly in the simplest possible way. All you need is charges and light and water. And if you go back to the clouds, these droplets have negative charge, so of course they will want to repel each other. However, the atmosphere has positive charge, and if you get the right amount of positive charge, they come together and you get a discrete cloud, and that's why you get something like this. How about biology? Sorry, I see my timer, my conscience over there. So does biology make use of radiant energy? Well, if you think about a cell, so the cell has got a protein in it. Of course, they're extremely crowded. I haven't shown it here. And next to each protein is a layer of EZ water. So these are negatively charged, and out here are positive charges. And so essentially what you have is separated charges that drive reactions. All the reactions that go on in your cells, they're either oxidation or reduction reactions. And and the charges are already there. They come essentially free. This is sort of like a proton pump, if some of you know biology. Now, what about larger scale use of radiant energy? We're absorbing this energy all the time, and the question is, do we just throw it away, or do we actually make use of this energy? And I'd like to suggest to you that we have a vascular system, and we have lots of capillaries that are all over the place. Many of them are superficial, but actually the light penetrates pretty deeply. If I turn off all the lights and put a flashlight here, I can actually see it through my hand. And so some of the wavelengths penetrate deeply in your body and my body, and the question is, can we use some of this energy just the way I've shown you? And I think it's possible because, so this is an image of the microcirculation, right? And this is muscle tissue. It's got these striations, and you can see the capillaries here. See this guy struggling to get through, and the reason is that the capillaries are smaller than the red cells, so the red cells have to squeeze to get through. So it's a big job for those red blood cells to make it through, and they could use all the help you can get. My Russian colleagues calculated that if the heart were responsible for this, pushing through these very small vessels, certainly the big vessels for sure, but these small vessels, it would need to generate 1 million times the pressure that it actually generates. So help is needed. So now we come to, well, gee, we already found that in these tubes, which are similar to blood vessels, we know that radiant energy, light, effectively drives this flow. And so the question is, might radiant energy help to drive flow in your capillaries, the small vessels? And I think it's possible. And just recently, an Israeli group published something we came across, and they found something interesting. They put, it was just one paragraph in their paper. I think it was the Weizmann Institute. They found that they killed mice. They clamped the main vessel, the aorta. The mice died promptly, as expected. However, using an optical technique, they measured blood flow in capillaries. It kept going for 1 hour. They did 10 experiments, and they found at least 1 hour continuing flow in every experiment. It was just one single paragraph, but they're now trying to figure out what goes on. I think what goes on is that the light that's coming in is driving the flow in the same way that we found in the laboratory. So basically, I'm suggesting to you that, nature created photosynthesis, used light, and I think if I were Mother Nature, I wouldn't give up exactly on this photosynthetic process as I created humans or animals. Why not use it? And so I think that the first step of photosynthesis, which is the splitting of water from light, is something that we actually use ourselves. Last 3 slides. Practical uses of this. Well, maybe you don't consider driving blood flow practical, but there are some practical things we've been following up, and that is, you have a surface, and I've shown you this so many times, and the question is, if you put one electrode here and one electrode here, you get energy out of it. We've been able to get energy enough to light a small display. And I think it's possible. This needs a lot of engineering development. It's surprisingly formidable. We're working on it now to see if you can get energy from sunlight and water without depleting the earth of its valuable resources. And the second one is getting drinking water. Well, it's no problem in Holland, but some places in the Middle East, for example, water is an issue. So I've shown you that if you have a hydrophilic material in water, and if the water originally contains bacteria and other junk that you want to get rid of, right, we have the exclusion zone here, and all the excluded stuff is here. So basically, if you could collect this material, this water from the exclusion zone, then you could get drinkable water, in fact, from ocean water. We think we can do that because we think that salt is also excluded, but we're not absolutely sure. So this is this separation now doesn't need a physical filter of any sort. It's driven by light from the sun, so it's literally free. This is another thing that's going on in terms of practical development. So I conclude by saying what I've shown you is that I think that the idea that water contains 3 phases that we all learned about is not correct. There's a 4th phase, and this 4th phase creates a new body of understanding that lets us really understand all these paradoxes and ununderstandable things about water. Now we can begin to understand them. And the second point is that water is a transducer of energy. You can't separate water from energy. Water is always absorbing energy from the environment and transducing it into mechanical and other kinds of work that I haven't had a chance to describe to you. And all of this is described in the new book that I wrote and just came out a couple of months ago. And if any of you are interested, it's you can't see it here, but it's evenerandsons.com. There you can actually read free without buying. You can read some of the chapters. You can't see it. I have a few flyers I'll just leave here if anybody's interested. And I think I'll stop here. So,
we had a 5-minute technical delay, so we have a little bit extra time here. It's such an appropriate talk for the UNESCO Institute for Water. I really want to thank you. You did a great job.
Thank you.
How about taking 2 questions, all right? First 2 hands I see. Hey. Okay, over there, sir.
So this is a liquid crystal-like structure. Have you looked at phase transition properties? Have you looked at whether by creating different geometries you can create different structures, temperature dependence, all the sorts of usual things to do with
well,
phases?
yeah, I mean, effectively, that was the main message of the previous book, that phase transition in your cells and my cells, that the water, this liquid crystal water, we didn't know at that time, 10, 12 years ago, its structure. But the main theme of the whole book was that this water undergoes a phase transition from this ordered regime to a disordered regime. And that now it's become even clearer that this is amazing because, are you a biologist?
I'm a theoretical physicist.
Yeah, okay, well, you know, your cells are negatively charged. Right, okay, so why are your cells negatively charged? Well, the classical explanation has something to do with the membrane, with pumps and channels and what have you. But actually, when you realize the cell is packed with negatively charged water, it's very easy to understand why the cell is negatively charged. It's the water. That explains also, for example, why gels made of proteins or polymers negatively charged. You stick an electrode in, you get the same negative potential, and gels have no membrane. So it's not due to the membrane, it's due to something inside the cell, and I think it's the water. Now, with the water undergoes a phase transition from this ordered, negatively charged liquid crystalline water to bulk water, suddenly the electrical potential goes from negative to 0. Then you have an action potential. Some of you know about action potentials. So there are many things that fall out so neatly out of these findings, and one of them, indeed, is the phase transition that can occur.
Yeah, we have one over here.
Yeah, so I was struck by both of your surfaces, the polyacrylamide, which the acetic acid, the carboxylate groups are acidic, and sulfonic acid, which is also, that's a source of negative. I'm wondering if this could be related to the Helmholtz double layer at the surface, where you get natural ionization of a polymer and double layer.
Good question. Yeah, so the problem is that the double layer theory that has pervaded all of physical chemistry, this layer exists for a few nanometers, and we're talking about hundreds of micrometers. So we're talking about a vastly different scale. And when we measure the electrical potential, we go from far away right to the surface. We never see this inversion at all. So if, in other words, the electrical potential is monotonic, it goes negative, negative, all the way into the gel or into the napkin, it's still negative. We never see a double layer. So if our results are right, double layer theory is wrong.
Professor, thank you. Brilliant.